Thank you very much for the introduction and for the invitation. It's a pleasure to share this moment with you. So I'm sorry I can't share my screen, so I'm gonna have to ask somebody to move on with the slides. So yeah, this talk is about phylogenetic conflict, and so I should probably first define what it is. I'm using here a very empirical definition. So what I'm calling a conflict is the situation where gene trees differ in topology from each other. So next slide. If we suppose we've sampled a set of genes in a set of species, and for each gene, we've built a tree, as we see in the next slide. If we're very lucky, all the gene trees will share the exact same topology, but this doesn't often happen, and most of the time what we see is that some gene trees like number 3 and 5 here actually differ in topology from the consensus, and this is what I'm calling a conflict. And next slide. The question I would like to ask today are, what are the causes of phylogenetic conflict, and what can we learn by studying them? So I probably should first recall that one major or important cause of phylogenetic conflict is reconstruction error. It might be that a gene tree differs from the species tree simply because we failed to correctly recover the true topology. And so there are a number of well-documented problems with pipelines for building trees, like you can fail at correctly calling orthologs, you can fail at aligning sequences, you can fail at specifying the substitution model. And for all these reasons, a tree can be an observed tree can be different from the true tree. So I happen to have done my PhD in the field of molecular phylogenetics a long time ago, by the end of the 20th century. And at that time, most of the discussions about phylogenetic conflict were about these methodological issues. So in the next slide, we see a sample article from this old times where the authors analyzed 15 genes in 4 species of mammals. That was a very big data set at that time. And they uncovered a large amount of conflicts with all 3 possible topologies being supported by some of the genes. And they concluded that maybe the guinea pig is not a rodent. So this paper triggered a large amount of discussions on the methods involved. And today we know that the guinea pig is a perfectly valid rodent, and that most of this conflict actually was due to so-called long branch attraction phenomenon. This is just an illustration of these old times. Then we entered the genomic era, and with it came the possibility of sequencing full genomes in closely related species. And that changes much the picture, because when you have full genomes in closely related species, many of the problems I was mentioning in the slide before, essentially, these problems are mitigated. Calling orthologs is easier with full genomes. Aligning sequences or building trees is easier with similar sequences. So we would have expected the observed conflict, to actually kind of vanish. But this is not what happened. And in the next slide, we see another paper from, if you can move to the next slide. Did I lose people?
Now we can hear. Are you still there, Nicolas? Now we can hear you again.
Can you hear me again?
You were frozen for some time, yeah.
Is that okay now?
Now it's okay.
Okay, so I was just saying that after this pre-genomic era, we entered the genomic era, and this changed much in terms of our ability to build correct gene trees. And yet, the observed conflict didn't really decrease. So in the next slide, can you hear me? Yes. In the next slide, this paper appeared 16 years after the previous one. And here the authors focus on the human, chimpanzee, gorilla triplet. And they report that even though a majority of the genome actually supports the classical topology grouping human and chimpanzee, the other 2 topologies also receive substantial support from a fraction of the genome. And this 30% discrepancy that we're seeing actually didn't look like being plausibly explained by reconstruction errors, given what we know of the method and the data. And this, I think, is when many of us, including I, started to seriously consider the other potential causes of conflict, which are the biological ones. So next slide, please. So it could be that gene trees genuinely differ from the species tree for biological reasons, and we know of two processes susceptible to explain such discrepancies. One is incomplete lineage sorting, or ILS, when the ancestral polymorphism interferes with multiple events of speciation. And the other one is gene flow, when an allele introgresses from one species to another. And so, next slide. Yeah, sorry. And so during maybe the last decade, there was a great deal of interest in trying to study and to distinguish between these two processes. And so here are a number of papers in a variety of taxa, having analyzed phylogenomic data set and analyzed the conflict in order to study these two processes. And one big lesson we can, I think, draw from this body of literature is that gene flow is more common than previously thought. If you can move to the next slide. Yeah, so I'm here highlighting the fact that, sorry, can you come back? Highlighting the fact that, yeah, all of this title actually insists on the prevalence of gene flow in these many taxa. So that's interesting. And another interesting result is that there are exceptions to this rule. And in the next slide, we see that in particular, apes are a notable exception. All or most or all of the papers, the many papers having analyzed the phylogenetic conflict in this group, only invoke ILS as the main cause of conflict in this taxon, which also is interesting and maybe is a little bit surprising. So from my viewpoint, to correctly interpret this body of literature I was reviewing briefly, one need to understand the details of the methods that we're using in these papers. And that's what the rest of this talk is about, the methods. So if you can move on. In particular, I would like to, yes, move on again. I would like to introduce APHID, which is a method I recently designed to distinguish between gene flow and ILS. I'd like to, next slide, to use it to maybe revisit some of the results I was showing just before. But before that, I thought I could maybe spend a couple of minutes trying to illustrate how exactly gene flow and ILS are expected to affect genealogies and what kind of signature they leave in the data. So to do this, in the next slide, we're gonna take a coalescent approach and try to think genealogies backward in times. And we will do that in the context, so this is called the multi-species coalescent kind of reasoning. And I'll do that in the context of the simple case of three species. So suppose we're interested in species A, B, and C. A and B have diverged T1 generations ago. A, B have diverged from C T2 generations ago. The effective population size, NA, is constant, and we've sampled exactly one gene copy, one allele if you want, per species. And now we're going to think about what kind of genealogy we can get out of this system. And we do that by simply tracing back the ancestors of the three samples we're having. So in the next slide, we see where were the ancestors some generations ago, and we can do that further back in time. And in the next slide, we've reached T1. And that's a key step, because from T1 on, now the A and B lineages belong to the same population, the same ancestral population, meaning that these two lineages, these two ancestors, at every generation now, they have a non-zero probability of being siblings, of being descending from the same ancestor. So next slide, maybe this didn't happen after this amount of time. And next slide, maybe this happened after that amount of time. And here, at that time point, A and B find their common ancestor in the ancestral AB population. They coalesce, and we're left with just two lineages. So we move on and until we reach T2, and then the same process applies again. And then in the next slide, we get the final gene tree for this specific locus we're looking at. So this is the normal situation, I would say, the one we expect to see most often, where the gene trees share the same topology as the species tree, AB versus C. We can note that the genealogical ancestor, the black dots are older than the speciation times. That's normal. That's reflect the existence of polymorphism in the ancestral populations. Another thing important to note is that this process of coalescence I was illustrating is actually stochastic. So the coalescence between A and B and between A, B, and C could have happened at different time points. For instance, in the next slide, we see another possibility, and in the next slide, yet another possibility. So if you move from one locus to another, you expect to get this kind of variance in branch length among gene trees, even though they share the same topology. Okay, this was for the normal situation. Now, an interesting situation happens if, in the next slide, if for some reason the A and B lineages haven't yet coalesced at time T2. But that's a possibility. You know, coalescence is stochastic. You have some probability to coalesce at every time point. You might well not have coalesced until T2. This is the definition of ILS. So incomplete lineage sorting, because it means that if you want the polymorphism that existed between A and B in the ancestral ABC population is still alive in the ancestral AB population and can be distributed to the descending A and B lineages. So what we have now is that the three lineages, A, B, and C, will start to coalesce together, if you want, in the ancestral population. If we're lucky, in the next slide, A and B coalesce first, and then, next slide, they coalesce with C, and we get a gene tree that, again, share the same topology as the species tree. But this only happens with probability 1/3. Because with the same probability, next slide, what happens is that B and C coalesce first, and we get another topology. And it could also be A and C that coalesce first, and we get yet another topology. So this is why this is how ILS is expected to generate some conflict among gene trees, with this ancient polymorphism being kind of randomly distributed across the A, B, and C populations. So how likely is this process to happen? Well, this is determined by essentially two parameters. One is the length of the internal branch, so the difference between T2 and T1. If the branch is short, it is more likely that A and B will not coalesce at T2. And the other one is the effective population size. And that's because N determines the coalescence rate. In small populations, you're more likely that two random individuals are related. So coalescence is faster in small populations, and it's slower in large populations. So you expect ILS to be more prevalent in large populations. And in the next slide, I'm showing an equation that very well approximates the probability for ILS for a certain value of T1, T2, and N. So that was the first process, ILS, maybe the less intuitive one. Now, so far we have supposed that the three populations we're looking at, A, B, and C, are closed population. But in nature, there could be some migration between populations and some reproduction between migrants and resident individuals. And if this happens, then it might be that some of the ancestors of one of the lineage could actually be a migrant. So if we look at the next slide, this is what I'm, so yeah, the next one, this is what I'm here illustrating. I am here supposing that this ancestor to lineage B actually was a migrant coming from population C. And this means that, thinking backwards in times, the B lineage moves towards the C population. And now B and C can start coalescing because they belong to the same population. And so if we go to the next slide, we see this happening. And again, next slide, we have this gene tree with a discordant topology due to gene flow, due to integration from C to B. In the next slide, we saw the other direction of migration, with a migration from B to C leading to yet another instance of a BC versus A topology with different branch length. It could also be, of course, migration from A to C, similarly. And in the next slide, one should keep in mind that you can also have, you can move to the next slide. We can also have migration between A and B, in which case we get a normal topology, AB versus C, but with a different set of branch length again. So yeah, I wanted to maybe, it's been a kind of a long time. I wanted people to see exactly what kind of pattern we expect, because this is what you need to feel if you want to understand your data correctly. And so now that we've seen these expectations, let's come back to our data analysis problem if you want. So in the next slide, what I'm showing is the situation. We have a species tree, and we sample many genes. We build gene trees. So in the next slide, we expect that most of the gene trees we built will share the correct AB versus C topology. But on the other hand, we will presumably also see the other topologies in the data set. Some of them in the next slide are due to gene flow. Some of them in the next slide are due to ILS, but we don't know which come from what process, and we would like to estimate the thing. We would like to quantify the prevalence of the two processes in the observed conflict. So how can we do that? Next slide. Yeah, that's what we want to do, distinguish between the two processes and quantify their prevalence. Probably the most popular approach to this problem in the next slide is the ABABABA method and its many derivatives, which basically consist in counting the number of gene trees with either of the two discordant topologies, BC versus A and AB versus AC versus B. This is informative because the ILS hypothesis predicts an equality between these two numbers, ILS being a symmetrical process. If ILS was the only determinant of the conflict, you would expect these two numbers to be roughly equal. So if you can detect a significant imbalance in the counts of these two topologies, you can safely claim having demonstrated the existence of gene flow in your system. So that's a cool way of proceeding. However, ABABABA has some weaknesses. ABABABA is good for detecting gene flow, but not for quantifying. And the reason is ABABABA can only detect the asymmetrical component of gene flow. So in the next slide, we'll see another example where, can you move on with the slides? Okay, maybe I'm not seeing everything. Yeah. So what I'm showing in this slide is that you have a situation where there's substantial gene flow both between A and C and between B and C. And so we have many gene trees, many discordant gene trees due to gene flow, but roughly the same amount for the two discordant topologies, because C is connected both to A and to B. And this is going to be entirely missed by ABABABA, even though gene flow is the dominant process in this specific case. So if you want to quantify, we need something else. Okay, so next slide. Let me try something. Yeah. Okay, so back to this figure, what we see from the top part of the slide is that besides topology counts, the gene flow and ILS hypothesis also make distinctive predictions regarding gene tree branch length. So with gene flow, what we have is a recent common ancestry between the two interacting lineages, so short branches, whereas ILS, in contrast, is by definition associated with very ancient coalescences and long branches. So a tall genealogy. And that's the beginning of an idea. If my discordant topologies, as we see in the next slide, if my discordant topologies are shorter than the canonical ones, then gene flow presumably dominates. But if they're taller, then ILS presumably dominates. And if we have a mixture of short and tall discordant topologies, maybe can we quantify the proportions? So branch length seemed to carry some signal that we might like to capture to distinguish between the two processes. And that's the idea behind AFID, which I'm now introducing in the next slide. So in AFID, we want to model gene trees as a mixture of scenarios where each scenario makes predictions on gene tree topology and branch length. So we have a no-event scenario shown here, where coalescences happen to any generations before splits. That's the expectation. And we get an AB versus C topology. This is the normal case. Then we have ILS scenarios. Next slide. So where the coalescences are older than T2, and we can get any of the three topologies with equal probabilities. And we also have gene flow scenarios in the next slide, where again, depending on who the donor, who the recipient, we can get any of the three topologies, this time with shortened branch length. So the model parameters include the effective population size, NE, the speciation times, T1 and T2, gene flow time, all measures in unit of mutation, which is what we can observe, and also parameters specifying the proportion of the mixture, parameters specifying the probability for a given locus to have experienced gene flow, so PAB, PAC, and PBC. The model also, the current implementation of the model also accounts for the variance in gene flow time among loci and the variance in mutation rate among loci, but I'm not going to develop on this. So in the next slide, I'm showing how basically we calculate the likelihood under AFID. So the model is this set of scenarios parameterized by the parameter I was just mentioning. And the data is the set of gene trees with branch length, rooted gene trees with branch length, and 3 species. And to calculate the likelihood, we first write in the next slide that that the likelihood of the whole data set is the product of the likelihood of every gene tree. Then the likelihood of a particular gene tree is written as a linear combination across scenarios. And finally, the likelihood of a gene tree given a scenario, well, this is 0 if the gene tree doesn't have the topology predicted by the scenario. And if it does, then we simply write the likelihood of a gene tree conditional of the scenario as product of Poisson deviates of mean equal, every branch length of the observed gene tree is seen as a Poisson deviate of mean equal to the expected branch length given the scenario. So we want to assign a high probability of a gene tree given a scenario if the gene tree resembles the expected branch length of that scenario. Okay, so I will skip, I think, the next 3 slides. If you can move, oh yes, let's skip this one and the next one and the next one. Yeah, and now, okay. And now that I've presented the method, I will just basically quickly show some of this behavior with simulations. So I performed simulations under the multi-species coalescent model with gene flow. So each simulated data set includes 2,000 loci. And in this simulation, the gene flow could happen at a constant rate across the whole phylogeny. So multiple events of gene flow were possible, and gene flow could be more ancient than T1, for instance. So the simulations depart the model assumption in several ways. So what we see is that despite these departures from its assumption, AFID estimates the parameters more or less reasonably. So the speciation times are a little bit underestimated. The effective population size is a little bit overestimated. But importantly, what we see in the next slide is that the relative prevalence of ILS in green and gene flow in red are reasonably well estimated, despite these small biases. So, and this was particularly true when the simulated conflict was not too high. So in this picture, the closed dots correspond to simulated data sets in which the conflict, the proportion of conflicting topologies was below 35%. Right. So this is not such an easy picture to get. And if you look at the next slide, you see the same kind of a result for another method that was published 5 years ago, I think, which is called Quibble. So this is the other method that explicitly aims at distinguishing ILS from gene flow. As you can see, the Quibble is very interesting theoretically. It focuses on the length of the internal branch of the observed genealogies. But yeah, with this kind of a simulated data set, it doesn't really perform well. Okay, finally, I decided to apply AFID to a couple of data sets, and with focusing in particular on primates. And so in the next slide, what I'm showing is an application of AFID to 3 data sets, 3 triplets of species in primates, using exon trees downloaded from the Orthomam database, so a highly curated database of coding sequence alignments in mammals. So first, I would like to ask you to concentrate on the size of the dots in this slide, which reflect the relative prevalence of gene flow and ILS to the conflict in these 3 data sets. And what I wanted to say here is that there's some kind of variation of the intensity of these 2 processes, but in all 3 cases, the 2 processes are substantial and significant. So it's not like if it's gene flow or ILS explaining the conflicts, it's usually both processes at the same time. Now, in this slide, the y-axis is the imbalance in topology count between the 2 discordant topologies. So that's the signal captured by ABABABA. As you can see, with the green dots, the gene trees that AFID considers are due to ILS have no imbalance. We have 50-50 of the 2 possible discordant topologies as accepted for ILS, which is symmetrical. However, if you look at the macaque dataset, we see that the gene trees that AFID predict to be due to gene flow, they have a highly significant and elevated imbalance with one of the 2 topologies, which actually is the one grouping the 2 Indonesian taxa in this case, is much more prevalent, much more frequent than the other one, which makes sense if gene flow is specific to one of the 2 lineages, so the classical way of thinking of it. And that's consistent with the literature. So the black squares in this slide correspond to the overall ABABABA signal, combining both the gene flow and ILS kind of topologies. So what we see is that by separating short from tall topologies, AFID did a better job than ABABABA of characterizing the whole process. Now, interestingly, in the other 2 data sets, what we have is a substantial prevalence of gene flow according to AFID, but in the absence of topological imbalance. And that was particularly the case in apes, which is interesting. And this is why I've decided to analyze a little bit more this triplet. So if you move on with the slides, yes, and again. So what I've done is to analyze not only coding sequences, but also genome-wide alignments. And so that's a much bigger data set for this triplet of species, human, chimpanzee, and gorilla. And I've done that So I've analyzed the different chromosomes separately. And what you see here are histograms for the main parameters for chromosomes. So as you can see, the X chromosome differs a little bit from the autosomes, with less gene flow and less and a smaller theta for this chromosome, which is expected theoretically, but that makes sense. And in the next slide, we see that maybe the most important result, we have with the data set roughly an estimation that roughly 18% of the genome is in conflict with the classical topology due to ILS, but also roughly 10% of the genome is in conflict due to gene flow. And that's new and different from the existing literature in which gene flow was almost never invoked. So in the next slide, we see the global picture, given by APHID of the history and demography of apes. So the effective population size is estimated to be of the order of 60 or 70,000, which is kind of similar to estimates in current population from the species. The divergence, the speciations are estimated to have happened respectively 8 and 10 million years ago, which is, again, kind of plausible given what we know of the fossils. And APHID predicts that roughly 10% of the genome was introgressed between gorilla and humanoid chimpanzee shortly after the human chip split. That's the estimation. In the next slide, I've added here. Can you show the next slide? Are you here? Yeah, sorry, no, the previous one, sorry. Yeah, so in this one, what I'm showing is what I'm showing in blue are estimates from another method, which is the Coal-HMM method, which has been used many times on this kind of data set. So the Coal-HMM doesn't consider gene flow, hence the 0%. And what it estimates is a more recent speciation times, sometimes unplausible given the fossils, and also a much larger effective population size, which also seems a little bit surprising. So this was just to say that by adding gene flow to the picture, we not only get a much better fit, high likelihood of the data, but we also get parameter estimate that makes maybe more sense than without gene flow. Okay, I'm close to finishing. So in the next slide, I'm recalling some of the assumptions and simplifications made by this method. It is very important to keep in mind that APHID is a heuristic. It's not a proper population genetic model, if you want. And that's because in APHID, we have a limited number of fixed scenarios in which branch lengths are arbitrarily fixed to their expectations, given these scenarios. But in reality, we have a much broader set of possibilities from the genealogies, given the stochasticity of the coalescence process, as I was showing. So that's an approximation that APHID does. The simulation suggests that it is reasonably robust to this problem, which is very good because these approximations make the method extremely fast. So you can analyze thousands of gene trees in a couple of seconds or a couple of minutes if you want confidence intervals. So that's much faster than the full coalescent method that also exists, right? And in the next slide, I'm also recalling that we have the important assumption that all the gene trees are assumed to be derived from non-recombining independent loci. So all the gene trees are expected to be independent, and importantly, gene trees are assumed to be reconstructed based on a non-recombinant piece of genome, and that's a very not an easy assumption to meet in a way. In the next slide, I'm mentioning a number of alternative methods, interesting methods. We've seen some of them in the talk before that relax some of these assumptions, but have sometimes also their own limitations. I think this is a lively field of research, and I expect it to continue to progress in the forthcoming years. That's a very interesting, combination of phylogenetic and population genetics. So not an easy part of the, anyway, not an easy task to do, but an interesting one. And finally, I would like to mention that there's another important assumption in the next slide, which is common to all these methods, which is that we neglect the possibility of integration from ghost lineages. So we assume that the gene flow we see is between lineages we've sampled, but there could be also contribution of lineages we don't see because they were extinct or because we didn't sample them that could contribute genetic material to the species we're looking at, and that's maybe that could be a major source of problem in this kind of analysis that so far I think has been neglected. And this is pretty much everything I wanted to say about this project. So in the next slide, I'm thanking all the persons mentioned here. If you want to try the program, please take a look at this website. And in the next slide to finish, I want just to emphasize that this work was published in the Peer Community Journal, which is probably the most ethical way of publishing currently. I warmly recommend trying the PCI. And if you're interested in ethical publication, please take a look at this database on academia-friendly journals in ecology and evolution we're maintaining at Montpellier. And thanks much for your attention.